Uh, so this is a continuation of last video. So the chromatography. So especially chromatographic technique used to separate the amino acid mixture. Okay. So the principle of chromatography is that the chromatography contain the setup has three components, that is a stationary phase, mobile phase, and component for separation. So these are three terms non, deals with the chromatography technique. Okay. So the mobile phase is a that is mobile phase travels up the stationary phase due to capillary reaction. So I'm discussing a paper chromatography. Mobile phase is generally mixture of a non-polar organic solvent. Okay, so mobile phase is normally non-polar organic solvent, while a stationary phase is a polar solvent. So where it is held? So here in the paper chromatography, so in the paper chromatography, polar water molecule is held inside the void space of the cellulose, or that is paper. Okay, so you see here, this is a chromatographic strip. So this is 0 point. I will more you will know more about these things. Okay, that is initial spot. You will spot the the amino acid mixture. Shall say this technique is also used in the plants and the biology for other. So here I am discussing about the separation of amino acids. So the mixture is spotted here in this paper, and that is zero initially, and then this strip is put into the a chromatogram, say a chromatogram, that is called a chromatogram, and as time goes, the all mixture will go up. So you can say that solvent is reached here. Okay, solvent is reached here, and the depending on the ability of how the polar the amino acids interact with the the solvent. Solvent will move. So according to that, the separation is done. So procedure I told you, that is you take a paper. Okay, that is normally uh, say like what do you say, paper of filter paper, like that strip of filter paper, and you made a mark, and in this spot, so this is your solvent in the beaker. Okay, this here the solvent is dipped, so solvent will move up. Okay, so the beaker and the solvent in, so two components are separated here. The initially the A and the B, so I can say glycine and alanine, like that, anything. So these two components are separated here as the sorry. So the two components are separated like that. As time goes, more separation will take place. That's as the more solvent the solvent is moved up. Okay, so after that, you spray the ninhydrin on the strip of this paper after drying the strip after separation, and you will spot the amino acids. Okay, you will spot the amino acids. Different amino acids will have different color, like that. So for example, here you see the leucine, alanine, and the uh, glutamine. So most polar amino acids will held strongly with water. Near the, so least polar will so least polar will move up, like that. If you are taking as the polar in the polar solvent, like water in the strip, and non-polar is placed, like this is your origin, like that. So you can say what is the difference between TLC, that is what is a thin layer chromatography and paper chromatography. The here is silica gel or aluminum oxide as used to the to help the water molecule in this paper chromatography is so less least absorbent is used like that. This is all about the the chromatography. You may have some more detail from internet or from textbook. Okay, so at least you have this information. So this point, some of the point I have mentioned here. So you draw the picture of a chromatograph and procedure. Okay, and you have to submit it at the end of the chapter and lecture. Okay, next topic is peptide bond. So what is the peptide bond? You know that two amino acids, the protein contain different amino acids bonded together. So two different amino acids or same amino acids held by together by a bond that is called a peptide bond. How the peptide bond is formed? For example, the alanine, this is the alanine, okay, and this is glycine, right? H is there, this is there. So they form a peptide bond, means the one of the amino group of the glycine and the carboxylic group of the alanine, and it's called condensation. Water is removed on condensation, form an amide linkage. An amide linkage. Okay, an amide linkage. Okay, so glycyl alanine. This is a dipeptide. So what is the dipeptide? Two amino acids form a two amino acid form dipeptide. Okay, so this bond is C double bond or NH bond is called what? Peptide bond. This is peptide bond. So what is the peptide bond? You can explain with the help of this. Okay, on hydrolysis of this is a condensation of or dehydration of between two amino acids. Condensation is small molecule is removed due to the cause of reaction is called condensation. Okay, so here water molecule is removed. So uh, so dipeptide means they contain two amino acid. The amino acids are called dipeptides. So here you may find out. Let me tell you the each peptide or a protein or a the dipeptide, tripeptide, etc. They have a two end, that is amino end and carboxylic end. Okay, amino and the C and the N end. This sometimes this is normally the standard notation is that amino group is placed in the left direction and carboxylic is RHS is C terminal and N terminal. So like that. So similarly, tripeptide contain three amino acids. For tetrapeptide contain four amino acids. For example, this is also an amino acid. 1, 2, 3, 4, 5, 6. You can say that hexa peptides because it contain six amino acids, and this is histidine. This is the C terminal, and this is the N terminal. Okay, so what is the N terminal? Means NH2 group terminal, and another is carboxylic acid group terminal is called H terminal. Okay, I think many of you have this idea, so you may have come across. I'm just refreshing you. Okay, so now let us discuss about the protein structure. So I have described you what is the peptide bond, kind of peptide bond, kind of peptides, that is dipeptide, tripeptide, tetrapeptide, hexapeptide, etc. Dear students, you have to be very serious about the class because you will have examination soon. Okay, I'm somewhat quick, and I'm just giving few of the things you have to. And I know you have many other papers also, so take this time very serious and do your best. Okay. Well, many of you have not submitted my last assignment, so but you, I will check. Based on that you will get your internal mark, right? So what is the sequence? So you can say that the sequence of amino acids can be determined using the okay, you can also find out what are the amino acids present, like enzyme carboxylipeptides. What did this carboxylipeptide carboxylipeptides cleave the C terminal? So, for example, here this C terminal, this, okay, here it is broken. So here it will be COH. Okay, then this is broken. Here will be COH, like that. So the uh, so based on using the, for example, carboxylipeptidase, we can determine what are the different amino acids present in a protein, like that. Okay, so which cleavage, for example, N-terminal cleavage enzymes are also there available. Okay, so I'm just introducing one term, that is carboxylipeptidase. That is which cleavage, cleaves the C terminal of the protein or amino acids. Okay. And what is the primary structure of the protein? So what is the primary structure of the protein means the sequence of amino acid present in a peptide chain and the location of disulfide bridge is called primary structure. What are the amino acids present? Then sequence, line by line, what are the amino acids present? One by one amino acids present in a protein, and the location of disulfide are called, that is called. So for example, that is very basic information about the protein. For example, see, uh, what are the different amino acids present if you are giving the sequence line by line, not by the structure. You are just giving this sequence line by line, the one this one for example, let me show the picture. So for example, this is a amino acid, whatever it will, this is insulin. Okay, insulin. So insulin, this is not actually the picture of the, this is just a sequence. This is because of it, there is no space like this. That's what it is. So you can say that between the cysteine and cysteine, there is an disulfide link. That is this. If you have this information, which is this is called the primary structure of that particular protein. Okay. Okay. So this is two chains are there, and like that. This is the sequence of a chain start from the glycine at N terminal and end with as per some of the information. This is glycine is the N terminal, and this for this chain A, and aspargine is the carboxyl terminal. Like that, similarly, the like that information. So this is what primary structure of the protein. Okay, you are clear? I think you got it. So similarly, this is also oxytocin, muscle-induced labor. So this oxytocin is okay. This is also a protein or okay, peptide chains. So this is like this. This is not actually the molecular structure. This is just a sequence structure, and vasopressin, another like that. So you see the in the this is the primary structure of the. I am giving one more example. That's all. You don't need to learn any of this sequence like that. But you just if you are giving some example, it's well and good. Otherwise, you just that is enough. Okay. This is not mentioned in the syllabus. So I'm just taking one example so that you can have a good idea. That's what that's my intention. Okay. Really, I have mentioned earlier like that. Now, what is the secondary structure of the protein? Okay, so primary structure, you got it. So the secondary structure of the protein means it gives the information about the sequence of the amino acids disulfide plus hydrogen bond present in a the peptide chain or an protein is called a secondary structure of the protein. Okay, so that is it also gives the about the plus hydrogen where the hydrogen bond are present like that. That information is called a secondary structure of the protein. So how many hydrogen bonds are there? So that define the shape of the protein. Okay. Okay, secondary structure refers to local folding structure. Local folding that form with the peptide due to the interaction between the atom of the backbone. So just remember what is the backbone means. Backbone of the amino acids means side chain of the amino acids. Okay, right? Okay, and R group. Okay, second structure. So most common type of the secondary structure are alpha helix and the beta pleated sheet. Okay, alpha helix means I think you this sometimes this is also called a salt bridge because salt bridge means this is the involvement of the this is CO minus and the NH3. That is there is a hydrogen bond. There is a chance of hydrogen bond between this CO minus and NH3 plus like that. So along the hydrogen bonding, dispersion force and this overall shape of the conformation of the bond I will discuss in the tertiary structure like that. Okay. So this is a two chain hydrogen bond that is sheet. I am also providing one sheet which contain all information in detail. I am uploaded in your along with this video. You have to go through it. Okay. So and this is the alpha helix structure. Alpha helix means just like the curling. So the this that is because of the you can say that intermolecular hydrogen bond. Alpha helix structure is due to alpha helix spring, just like spring. Okay, say just like spring. Okay, so you see here this protein, this is a protein. It has some area it is an alpha helix structure, and in some other area it is a beta pleated sheet. Okay, beta pleated because of the interaction of the hydrogen bonding between two the peptide or two protein or peptide chain. Okay, this is because of the alpha helix, right? This alpha helix. Okay, so tertiary structure of the protein, it include that's all about the prime secondary structure. So you read from more in your that sheet I have provided you. Okay, and next is a tertiary structure of the protein. So, what is the tertiary structure of the protein? The tertiary structure of the protein, it includes weaker force of attraction, non-polar dispersion force between hydrophobic part of the same chain that widely spread by primary structure close to space. Intramolecular result from twisting and faulting. That is, if you know that information, that is also called twisting and faulting in the are also because of the uh, tertiary structure that gives you information, right, just like or non-covalent bond information. So the tertiary structure of the protein include sequence of amino acid, hydrogen bonding, and apart from that they include hydrogen bonding, ionic bonding, dipole interaction, London forces, etc. So these are hydrophilic interaction, hydrophobic interaction. So the London force is a general term that include the all London force means dipole interaction, induced dipole interaction. All information is there, which means that is the ternary structure of the protein. Okay, so for example, that gives the shape, that gives the information about the shape of the protein actually. Okay, so if you know all interaction, all, so there may be some cumulative effect, etc., like that. So R groups are repel one, some R group may repel, some R group may attract with each other. So these opposite charge ionic bond, okay, ionic bond similarity polar group can hydrogen bond or dipolar interaction also important, that gives hydrophobic interaction, hydrophilic interaction, all together information. Then that is called ternary structure of the protein. That gives the actually shape of the protein. And this is actually like that we see here. This for this many interactions may result there the all information. So you have ternary structure means you are predicted the exact shape of the protein. Okay, so I have limited knowledge of the biology. Okay, I'm sorry, but I'm talking in terms of chemistry point of view. Next is uh, so like that, collagen, fibrous protein, globular protein. So some I have some more classification are there. I will discuss in next class. I'm here only discussing about the structure. Okay. So hemoglobin, collagen. These are different. So so this you read on this. This is not a nothing is mentioned you in your syllabus. Your syllabus mentioned primary, secondary, tertiary, and coronary structure of the tertiary structure of the protein. Anyhow, if you know the ternary structure, then what is the coronary quaternary structure of the protein? Means suppose some amino acids, some protein may also contain multiple of the chains. Okay, if that is many proteins are made up of single polypeptide chain and have three levels of structure, that is primary, secondary, and tertiary. However, some proteins are made up of multiple polypeptide chain, also known as subunit, and these subunit together they give the quaternary structure of the protein. That is, in simply, I can say that hemoglobin has the quaternary structure, quaternary structure. Okay, because they contain multiple polypeptide chain. Okay, I am stopping here. So go for your assignment and submit. Thank you for watching. You can ask any question, any doubt personally to WhatsApp or any platform. Thank you for watching.